Hi. Okay, so this is going to be your equilibrium pre-lab. So I'm just going to kind of jump right into it. We are going to be looking at 4 different experiments for this lab. The first one is just going to kind of get us our sweat with the nature of equilibrium. So what does it actually mean for something to be in equilibrium? And then the other 3 are going to be, we're actually going to look at Le Chatelier's stressors on equilibrium systems to see what happens there. The first one's going to be concentration. The second one, so we're going to change concentrations of the different substances in the chemical reaction. The second one is we're going to experiment with temperature. So what does temperature have to do to the experiments? And then the last one we're going to look at is pressure and how that affects the equilibrium. So the purpose of this lab is to investigate different factors that may influence the balance of the equilibrium in a reversible reaction. So that's ultimately what we're doing, and we're going to be looking at 3 different things with that. Okay, so the first factor, so I'm going to call these factors, but basically they're just parts. So part A or factor A is actually just the nature of the equilibrium. So what we're going to be doing is we're going to be using an indicator, and if you remember what an indicator is what we use to tell us whether something is acidic or basic. So it changes color based off of how many hydrogen atoms are present, or hydronium, however you want to look at that. So an indicator loses or gains an H plus and changes color in the process. I kind of just said that. So we're going to need to know these 2 following equations to understand the reactions. The reason why I'm saying this is in order to tell whether there's a shift in the reaction, meaning am I on the product side or the reactant side, the product side and the reactant side have to have different colors. So in this case, this is our equation for the indicator. So hydrogen plus ion, which is the indicator. Most times indicators have these very elaborate chemical structures, so we just indicate them as IN. And ultimately what's happening is in equilibrium, if the indicator loses the hydrogen, that means it's changing color, and then that means that the indicator becomes the conjugate base of the starting acid. And then the second equation is our equilibrium that we use for water. Okay, so what is this actually going to look like? So what's going to happen is I'm going to start with water, and I'm going to add this indicator called bromcresol green, and then we're going to swirl record color. So this is just a procedure, so I'm going to go through this pretty quickly. We're going to add 3 drops of HCl, which we know anytime we're adding something like a strong acid, HCl, really all that we care about is the H because the Cl. So ultimately, if I'm adding HCl, I'm just adding H because the Cl is just a spectator ion. So we're going to record observations from that. We're then going to add sodium hydroxide to the solution, and then we're going to restore the original color. So basically, step 2 is I'm making the water acidic. Step 3 is I'm neutralizing that H plus with the hydroxide to get it back to its original color. So again, you'll notice I put sodium hydroxide, but like we've said before, sodium is just a spectator ion, so really all I'm adding is the hydroxide. So I'm making water acidic, adding base to make it neutral again. And then I'm going to continue to add hydroxide, which means I'm now making my solution basic. And then I'm going to add 0.1 molar H again until the solution reaches the transition color. And so ultimately, we're just kind of making observations about, so if I'm going from one color to another, that's indicating that there's a shift in that chemical reaction. So that's procedure A. Your data table will look something similar to this. We're ultimately disturbing the chemical reaction that's at equilibrium. So every disturbance requires an observation. So that's ultimately what that's going to look like. Okay, factor B. Now we're actually going to start talking about actual different Le Chatelier principles. So if we affect the concentration, so we're going to be using 2 different chemical reactions for this problem. The first one, you'll notice that I have ions here. You'll see them written sometimes as iron with something else, but that something else is always a spectator. So these are the net ionic equations, and so that's actually what matters to us. So iron plus 3 is yellow, thiocyanate or cyanide. Yeah, I was right the first time. Thiocyanate, which is SCN-1, is colorless. So then when we take the iron and the thiocyanate and put them together, that's where we get a red color. And then equation 4 is I have, again, iron is yellow, and then you have this H2PO4 minus. When we put those two together, it makes it colorless. Okay, so it's important that we know what's happening with these chemical reactions. Okay, so the procedure is going to look a little bit like this. We're going to add, it says potassium thiocyanate, but we know potassium is a spectator, so ultimately I'm just adding that that thiocyanate. We're going to transfer it into a Petri dish. This is actually a really cool reaction. We're going to add 5 drops of iron 3. It says nitrate, but again, nitrate's a spectator, so it doesn't really matter. All I really care about is the iron. I'm going to add it into different spots on the Petri dish, and we're going to record observations. I'm going to then swirl to get an orange color. I'm then going to add a pea-sized thiocyanate crystal in one spot. I'm going to wait 30 seconds, make observations. Swirl the solution to make it uniform. Add a small amount of that sodium phosphate monobasic, that's the H2PO4 minus, I think it is. You're going to wait about 60 seconds before making observations. Swirl until crystal dissolves. And then we're going to add more iron again, record observations. We're going to add more thiocyanate, record observations, and then we're going to clean the Petri dish. So ultimately, we're adding more substance. So every time we add more substance, we're adding more moles of that substance, which ultimately means we're making the concentration higher. So we're looking to see what's actually happening. And since there's color associated with each different chemical reaction, as the color changes, we know the shift in the chemical equation. So again, we're disturbing it, so therefore we need to make observations about it. Factor 3 is temperature. We're going to do 2 different reactions. Equation 3, we're going to use that one again with the iron and the thiocyanate, making iron thiocyanate, iron 3 thiocyanate, and then we're going to use this other chemical reaction where we're going to have these really pretty pink and blue colors. I'm going to kind of go through this quickly because you can always just pause it at the end to write the procedure. But we're going to start with 2 milliliters of cobalt chloride. We're going to use A as a control, right? You always need a control to make sure you know what's changing. We're going to add water to test tubes B, C. And then I'm going to add 3 drops of 12 molar, which is a really high concentration of H+, meaning it's very acidic, to test tube C. I'm going to place test tube B in the hot water bath for 2 to 3 minutes. So test tube B is going to be the hot one, which means test tube C is going to be the cold one. Ice, record. And then we're going to, so steps 1 through 6 is part 1 of procedure C. Part 7 through the end is part 2 of procedure C. So we're going to absorb initial colors. We're going to make a solution. We're going to do the same thing. We're going to label test tubes D through F. D is going to be control. And then we're going to add our 1 solution to E, ice water bath, our 2nd solution to F, hot water bath. So these are what your data tables are going to look like here. So you have A is the control, B is hot. So we're looking for a before color and an after color. C is cold, so we're looking for a before color and an after color. And then over here we have D, control. E is cold, before, after. F is hot, before, after. And then it asks us to record initial colors of iron 3 and thiocyanate, but we already know those colors, but that's okay. We're going to record them anyways. Iron is yellow, thiocyanate is colorless. Okay, then I'm going to go fast through this one. Effective pressure, we are going to use this reaction. We're going to use carbon dioxide, water, and then we're ultimately we're going to be using seltzer water for this one. So again, we're going to use the bromocresol indicator to tell us whether our pH is acidic or basic. So we're going to add 50 milliliters of seltzer water and 4 milliliters of the bromocresol green. And we're going to draw up 25 milliliters into a 140 milliliter syringe. We're going to record initial volume of liquid and gas. We're going to record pH. And then what we're going to do is we're going to expand. So we're going to, if I'm expanding it, we're going to talk, you have to figure out whether you're adding pressure or taking away, but I'm pretty sure you can, if you're expanding it, you're making it, you have less pressure. Shake until you see further color change. Compress to 45 milliliters. So you're making more pressure, and then you're gonna shake the syringe and then record the pressure. We're gonna do repeat steps 4 and 5, so we're gonna do 2 trials of that. So this is data table D. So you're gonna need to record initial volume, and then we'll have 100 milliliters pH 1, pH 2 for our 2 trials, 45 milliliters pH 1, pH 2. And that's it. And then once we do the lab, we'll talk about what the post lab is going to look like. Thank you.